Good morning, guys. I'm going to be posting one, if not two, videos this weekend. I'm going to try and combine a couple lectures together. I think I can do them pretty quickly. Full disclaimer, I'm not as knowledgeable in the upcoming topics as I was previous ones, like glycolysis, but people complimented me so much I'm willing to give it a good shot and share what I do know. So starting with glycogen. So I'm just going to use his PowerPoints. So just remember that the brain needs glucose. It can't use anything else, glucose only. So this is sometimes one of the many reasons why just eating, a slice of bread isn't enough energy to keep us going. Our brain needs lots of energy. It can't use, protein or fat for energy like the rest of our body can. So that's why we have additional processes besides just eating straight up glucose and being able to store it for future use and create it from other things. Just as a note, remember that glycogen is not the only source of, so we have glucose straight up as a source of glucose, duh. We have glycogen, which we're going to focus on first, where we can unpack it into glucose, and then we also have gluconeogenesis, which was from a separate lecture where we're actually completely reconstructing something else into glucose. So those are our three sources of glucose. We can get it from glycogen, gluconeogenesis, and of course, just glucose itself. I like this image. It kind of shows you how, if I eat food, this blue line right here, I'm going to spike up my glucose levels, which makes sense. I just added a bunch of glucose to my body. But as I start to run out of that dietary glucose I ate, I'm going to start sourcing glucose from other places. First, starting with glycogenolysis. We're going to break down our stored glucose, and then even later on, I can do a little bit of gluconeogenesis if I need to supplement that further. So maybe if I ate just bacon for breakfast, that protein in my bacon, if I don't have enough glucose or fats, can be remodeled into sugar for my brain to use. So we have two locations where we can store glycogen. Remember, glycogen is our packaging, our condensing of a lot of glucose molecules, so we can store it in the liver and we can store it in the muscle. Remember, you cannot store glycogen in the blood. It doesn't go in the blood. We only have just straight up glucose in the blood. So liver and muscle are where we can find glycogen. And remember that liver, in almost every process that we've talked about, is going to be the overachiever with anything that has to be shared. So yes, muscle does have glycogen. Any glycogen that's broken down is going to stay within the muscle. However, the liver has lots of glycogen too, and it's going to share its glycogen with the rest of the body. So if my brain needs extra energy and I've run out of straight up glucose, the glycogen in my liver can be broken down to supply other body systems, like the brain in my example. So this is what glycogen looks like. Think back deep and hard to your lectures on carbohydrates from Biochem 1, and you'll kind of remember what your different glycosidic linkages are, hopefully. So two big things when we're talking about glycosidic linkages are alpha and beta, and that's whether our carbon or our oxygens are sticking up or sticking down. They're sticking down in everything in our body's glycogen store, so we're always going to be dealing with alpha bonds. And then we have two other categories. We have 1,4 chains and we have 1,6 branches. So 1,4 chains, we can see here and here, they're making these, and also here, they're making these nice straight lines. Our 1,6 bonds are going to be our branches. So associate these 1,6 bonds with the branches off of the main chains. All right, so first we're going to talk about getting glucose into the liver. Maybe we need to make some glycogen. Maybe we have a lot of glucose in our blood. Where is it going to go? One of the places it can go is the liver. We're going to use the GLUT2 transporter to do that. Remember that glucose doesn't really like to just go through the cell's membrane. So we're going to need channels. Our channels or our transporters for glucose are always going to be called GLUT. And it just happens to be number 2 that can help glucose get into the liver. No matter what process we're doing, whether we're doing glycolysis or we're doing glycogenesis, we're going to start with this step. We're going to use, because it's the liver, glucokinase. Remember you saw hexokinase for glycolysis. This is a very similar enzyme. This one's just specific to the liver. And we're going to add a phosphate group to the 6th carbon on the glucose. We're going to make G6P. If this step doesn't look familiar to you, go back and review glycolysis. Because anytime we're going to enter a cell, we have to do this because this is what keeps the glucose inside the cell. Or it's just going to go right back outside of the liver through the GLUT2 transporter and it's not going to be able to do anything. Just to let you know, we're going to see more activity from this and this, well, mostly this. We're going to see more of this activity when we eat lots of carbohydrates. Because remember, if we have more than we need to store it. Where are we going to store it? We're going to store it in the liver. If we're storing into the liver, we're going to get into the liver, and then we're going to have to undergo this reaction to keep it in the liver. So we're going to obviously see more of this when we have lots of glucose, like after a high carbohydrate meal. So moving on specifically to glycogenesis, remember glyco is going to refer to glycogen and genesis is going to refer to the making of. Pay attention on the exam to what words you're reading. We're getting into the point where we have lots of words that look kind of similar but mean different things. So definitely break apart those words. Make sure you know what you're talking about. I'm making glycogen. So a couple enzymes here that we're going to use. These are definitely things I think you're probably going to need to have at least somewhat memorized. We're going to talk about each little step. So the first is the hexokinase glucokinase step. That's what we just talked about. We added the phosphate group to our glucose and we trapped the glucose in the cell. The next is phosphoglucomutase. So mutase, I said in glycolysis that if you see mutase, think move. Move. And that's going to hold true again. What we're doing is we're taking this phosphate group and we're moving it, move, to carbon number 1. So now instead of glucose 6-phosphate, I have glucose 1-phosphate because the phosphate is on carbon 1. Next step is UDP glucophosphorylase. So UDP, if you see a UDP, you know you have to be in glycogenesis. That's the only place we've talked about it so far. UDP is essentially like a marker or a tag or some sort of identification molecule. We're essentially sticking a UDP on this carbohydrate so the body knows I'm going to be storing this. It's a name tag essentially. So instead of a phosphate group, we're going to replace that phosphate group with uridine diphosphate glucose. That's UDP. You can see that right there. So first we got it into the cell, we trapped it, we moved it, and now we've marked it. Next, now that we have it marked, we can actually start kind of making up our glycogen molecule. What we're going to do is we're going to add another glucose on this UDP side. So I took this, I did another one of those, and I stuck this one here. So that's what you're seeing right here. So important to note, because we're just making a normal chain right now with glycogenin, I'm making an alpha 1,4 bond. So I'm just adding 2 of these together to make this. So that's that tag on the end. Okay, so that was glycogenin. Next is glycogen synthase. So essentially it's going to take what we did before and we're going to just keep going. See how I had this long chain drawn, all 1,4 bonds though. So we're really starting to synthesize glycogen by making these long chains. We're just adding more glucose molecules. Yes, there will still be this UDP at the end here. And then our last step is the branching enzyme, because remember, if we look back here, we said that glycogen has these 1,4 chains, but it also has these 1,6 branches. How do we add those 1,6 branches? With branching enzyme. Names are going to sometimes be really easy. So essentially, every 7, this is not accurate, I didn't count, but removing a 7 glucose chain, so every 7 glucoses, I'm going to form a 1,6 bond so that I can have a chain. And that's glycogen. Of course, every process in our body has to be regulated. We talk a lot about hormone regulation. The good news is when it comes to anything carbohydrate related, we're going to be working with the same hormones, and the rules for those hormones are going to remain the same. They can just act on more than one process. Insulin is always going to lower our blood sugar. So you have to ask yourself, okay, if I'm dealing with insulin, do I need to turn on or turn off this process to lower blood sugar? So insulin, if it's going to lower blood sugar, we're actually going to upregulate glycogenesis. We're storing it. We're taking it out of the blood and we're packaging it up. So that's going to be upregulated by insulin. So here you can see insulin specifically activates glycogen synthase, which was right here. So if this is upregulated, we're making more glycogen. It's also going to stimulate more uptake, and it's going to create more transporters for glucose into tissues. So insulin, more glycogenesis. Glucagon is always going to be the opposite of insulin. So if we have glucagon, we're going to try and increase our blood sugar. So why would I want to take it out of the blood and store it? Exactly. So we're going to down-regulate glycogenesis with glucagon. It's going to do the exact opposite of insulin, which is going to stimulate a different process at the same time. This process is going to increase blood sugar. This is glycogenolysis. Glycogen. Lysis means cutting or breaking or chopping up. So I'm breaking up glycogen so I can release those free glucose molecules into the blood. I like this picture for diagramming glycogenolysis. This is our glucose molecule, or our glycogen molecule. You can see here they're demonstrating in red alpha 1,4 bonds. Those are our straight chains. And in the green, they're showing us one of those branches, one of those 1,6 bonds. So just a few enzymes to break down glycogen. The first is phosphorylase or glycogen phosphorylase. It can just chop away at these 1,4 bonds. It can do it here and it can do it here. It doesn't really matter. But you're noticing that these chains here got a lot shorter with glycogen phosphorylase. So now we're going to have two debranching enzymes, and here's why. If I'm Mr. Pac-Man and I'm chomping away at these glucose molecules, by the time I get down to like here, I'll get more like the picture, we're going to run into a problem, and that problem is this really tiny little angle between the branch and the main chain. And unfortunately, glycogen phosphorylase is extremely limited in two ways. One, it can only break 1,4 bonds. This is a 1,6 bond, and it's also really narrow, and it doesn't fit in here. So our first debranching enzyme, I want you to think of it making more space. It's going to take groups of 3 off of this branch, and it's going to stick them onto the main branch. And that's what we got over here. These highlighted ones are now on the main branch, and we're just left with this singular 1,6 bond. So this is glucogen transferase. It's essentially chopping off the last little bit of the branch until we get to that 1,6 bond. That leaves plenty of room for the third enzyme, and that's 1,6-glucosidase, which is just another debranching enzyme, and that one is specialized in cleaving these 1,6 bonds. And you'll see, after we cleave that 1,6 bond, we're left with a nice straight long chain, and of course, glycogen phosphorylase can just finish the rest of this off. Same things that I said here. Make sure you know which ones can break 1,4 linkages and which ones can break 1,6 linkages. These are talking about hormonal control. We already talked about insulin and glucagon when we were talking about glycogenolysis. We said that insulin was going to increase glycogenolysis. It's going to decrease or inactivate glycogenolysis. These things are always going to be like working in opposition to each other. We're also going to see epinephrine frequently in these next couple of lectures. Epinephrine and glucagon are best friends. They tend to work together. Just know that epinephrine, the main difference is, of course, where it's produced, and that it's also released in response to stress and hypoglycemia, where glucagon is only hypoglycemia. And then cortisol, that's long-term effects. Chronic is long-term. If I am stressed out a lot, and trust me I am, I'm going to have lots of cortisol, and I'm going to have lots of activation of glycogenolysis. So definitely, he even said himself, if there's a table, know this. Alright, so we said that we can have glycogen in the liver, and we have it in the muscle. Remember, the muscle does not share, the liver does. So muscle is especially going to be the one activated by epinephrine, and remember, I think we all are pretty confident what epinephrine is, what it's released in response to. This is our fight or flight response. So when Dr. Tomodachi gives me a neuro exam and I start hyperventilating, I'm releasing lots of epinephrine. Epinephrine is going to go to my muscle, and my muscles are going to start breaking down their glycogen. Alright, and then, so we talked about hormone control, but there's other ways we can control processes in cells that are not hormones. So he gives us 2 ones to focus on. Influx of calcium ions. There's one reason I wanted to wait to do this lecture. I wanted to wait until we were done with the nervous system in neuro, because we're studying what happens in the muscle to make it contract, and hopefully you're going to remember, as I mentioned this, that calcium ions are released from the sarcoplasmic reticulum in response to nerve impulses. So our nerves say, hey, muscle, contract. It's going to do this by actually releasing calcium in that muscle, and the calcium is going to cause the muscle to contract. So calcium equals contraction. Contraction requires energy. So if I need energy, I need more glycogenolysis. I need to break down my glycogen for glucose to be used for energy. So we can see that it's going to activate this enzyme called phosphorylase kinase, and we're going to come back to that. Just tag that in your brain for a minute. The other way we can regulate this process without hormones is increased AMP. We're used to seeing ATP, adenosine triphosphate. If I break a phosphate group, we get ADP, adenosine diphosphate, and AMP is adenosine monophosphate. Mono means one. We only have one phosphate group left. Oops, what happened? So if you see AMP, think that your body is desperate for energy, because we've used that ATP like 2 times already. So if I have this alarm signal that I need more energy, I need to speed up this process. I need to break down glycogen, I need to get my glucose from it, and I need to do some glycolysis. So it's going to, just like this calcium ion example, activate this phosphorylase kinase, which we're coming back to. It's also going to relieve inhibition of PFK1. Remember, PFK1 was from glycolysis. That was like the 3rd step of glycolysis. So if that's inhibited, we're not breaking down glucose for energy. So if I'm relieving the inhibition, I'm disinhibiting it, I'm turning it back on, we're going to increase glycolysis, we're going to get more energy. All right. So let's address the elephant in the room finally. What the heck is phosphorylase kinase? Phosphorylase kinase is an enzyme that's going to regulate other enzymes. So we talked about glycogen phosphorylase, right? Going all the way back, this is where we saw it. It was breaking those alpha-1,4 bonds. But there's actually two forms of it, B and A. One is inactive, one is active. It's always a good thing in our bodies when we have two different forms of things so we can turn them off when we don't need them. But, let's say I haven't eaten in a while, I need to start breaking down glycogen for energy. I need to turn it on. What's going to turn my glycogen phosphorylase on? This phosphorylase kinase. So, going back to these, if I'm activating phosphorylase kinase in response to these conditions, I'm going to make B to A. I'm going to go from inactive to active, and therefore it's activating glycogenolysis. So when we read these slides, when we read activates phosphorylase kinase, right in parentheses, glycogenolysis. Increased activity of this equals more of that, just in a roundabout way. So that is it for glycogen. Moving on to gluconeogenesis. So remember in glycolysis, we did all these 10 steps and we made pyruvate, and pyruvate was converted into acetyl-CoA using that multi-enzyme complex. And that went on to the TCA cycle, and we did the ETC, we produced lots of energy. It was all good and dandy, but have you ever been exercising and you're going for a run and then you start gasping for air, your muscles start burning? It's because you don't have enough oxygen to do that much of the electron transport chain. And our body has a backup system, and that is lactate. Glycolysis is super fast, super easy to do. Our body can do this as easily as I can snap my fingers rapidly. So the electron transport chain, the TCA cycle, take a lot more time. We're going to want to do those. They're going to produce more energy, but sometimes we don't have time. We need to undergo a lot of glycolysis. Boom, 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 boom. But I don't have time to make that pyruvate into acetyl-CoA. I don't have time to put it through the electron transport chain. I don't have the oxygen needed to put it through the electron transport chain. So we're going to do this backup system. We're going to take pyruvate. We're going to use an enzyme called lactate dehydrogenase, and we're going to make it into lactate. This is good for two reasons. I get rid of my pyruvate. I make it into something else. This something else we'll worry about in a few minutes when we get into gluconeogenesis. It's also important for recycling this NADH. Remember in glycolysis, after we made DHAP and glycerol 3-phosphate, we had to use a dehydrogenase, and we made NADH, and we said that's really awesome because that goes onto the electron transport chain. Well, it's not going onto the electron transport chain anymore. We don't have enough oxygen to use it. But I can't just have a buildup of NADH in our cells. So not only is this enzyme converting our pyruvate into lactate, the real purpose of it is actually to get us back to NAD+. That can go back and do more glycolysis. But now I have my lactate. And what the heck do we do with lactate? It burns our muscles. It's not really good to have in high contents in our body. We're going to recycle it. This is how we're going to do it. We're going to get it out of the muscle where we found it and we made it, and we're going to undergo the Cori cycle. So here we see our lactate. So what we see in here in the muscle, we have our glucose. Yum. We're undergoing glycolysis. We made that NADH, and we got 2 pyruvate. Uh-oh, electron transport chain is shut down. what do? We're going to use LDH. We're going to recycle that NADH back into NAD to go back here. And I'm going to make this lactate. Lactate is useless in our muscles. We need to go somewhere else to use it in a different way. Lactate is going to go out of the muscle into the blood and then into the liver, where it's going to again use LDH, lactate dehydrogenase, to get back to pyruvate. And you're going to say, Rachel, we just did this to get back to here. Why on earth did we do that? Pyruvate can't cross out of the muscle and into the blood and into the liver. Lactate can. So if we ever do something, transform it into something, move it, and transform it back, it's because this molecule that we started with can't do the moving, but the other one can. So as we move into things like the malate shuttle, it's the same reasoning. We can't get that one molecule out into the blood for transport, so I have to convert it into something that can. From here, once we get our lactate into our liver, and we convert it back to pyruvate using lactate dehydrogenase, I can undergo this process that we're about to talk about called gluconeogenesis to make it into more glucose. I can use that glucose again for energy. That's why this is a cycle. This is the Cori cycle. So, gluconeogenesis, definition here. Remember, we're making glucose, so glucogenesis, and neo means new, and that's because we're going to make it from things that aren't carbohydrate sources. So we talked about breaking down glycogen into glucose. That's not the same thing as gluconeogenesis because it was already a carbohydrate. Now we're taking our fats, we're taking our proteins, we're taking our pyruvates, and we're making them into glucose. So that's why it's gluconeogenesis. And we have two organs that can do that. We have the liver and the kidneys. And remember, we said that the liver was the star child, the prodigy. It does most of the work. The kidneys kind of are selfish, and we'll talk about that more later. Something to make sure you have memorized for the exam is the location of these enzymes. We're going to talk about a couple enzymes that are unique to gluconeogenesis. A lot of the enzymes in gluconeogenesis are just the same enzymes from glycolysis. We're just going backwards. But remember, we had those irreversible steps. When we have irreversible steps, we're going to need a new special enzyme to overcome those steps. And just location is important, it is relevant, so make sure you have that kind of memorized, keep that in the back of your head, so as we talk about these guys, we know kind of where they are, because it is kind of relevant. Alright, where to start? Gluconeogenesis. We've used all of our raw glucose, we've used up all of our glycogen, now we got to make some more. This is kind of another last-ditch effort. This is going to require some energy, but remember, we're forming glucose from non-carbohydrate sources. We had our lactic acid that we talked about a minute ago, we have proteins, and we have glycerol, which is going to come from fats. So these are our new enzymes. If I want you to kind of pull out a diagram of glycolysis to reference. We are going to follow glycolysis more or less backwards through the process. If I do not address a step, it's because it's the same enzyme, it's just a reverse reaction from glycolysis. Pretty much, these are overcoming, this is kind of special, we'll talk about that. These guys work together to overcome step 10, this one overcomes step 3, and this one overcomes step 1 of glycolysis. These were our 3 irreversible steps. Everything else stays the same. So, pyruvate carboxylase. What does it do? We're going to make pyruvate into oxaloacetate, or OAA. So, remember, we said that we got this pyruvate into the liver. So we're in the liver now. Remember, we also said that this enzyme is in the mitochondria. This makes sense, right? We take any pyruvate we have, we're always putting it in the mitochondria. But I don't want it to stay in the mitochondria. I can't use it for the electron transport chain anymore, so I got to get it out of the mitochondria. How am I going to do that? This enzyme is going to help us. This enzyme is going to help us start this malate shuttle I mentioned earlier. So essentially what pyruvate carboxylase does is it takes a pyruvate and something called a carboxybiotin. So what we're looking at here is a biotin molecule, that's one of our vitamins, and a carbon dioxide. Carbon dioxide plus biotin equals carboxybiotin. And I'm going to mix them over. Essentially this biotin, I like to think of it carrying the carbon dioxide over to pyruvate. That leaves us with biotin. And OAA is what happens when I add this carbon dioxide to pyruvate. Yay, now I have OAA. This is kind of more or less our first step of gluconeogenesis, getting that pyruvate into something else, because I can't really use pyruvate for much unless I can undergo the electron transport chain, which isn't an option if I'm doing this. So now that I have OAA, I need to get, my goal right now, I said, was getting out of the mitochondria into the cytosol. OAA is great for going through the malate shuttle. Malate shuttle is when we take OAA, we convert it to malate. Malate goes out of the mitochondria and goes back to OAA. And once again, we said this with lactate. If I'm taking something, converting it, moving it, and converting it back, it's because this molecule is the one that can transport through the cell. So this is how we get to OAA. This is how we get it out of the cell. Now that I have OAA, I'm really going to start actually forming the glucose molecule. This is kind of all like transportation related processes. Now we're really going to start the actual building of a glucose molecule. And the first enzyme that helps us do this is PepCK, or phosphoenolpyruvate carboxylase. And remember, this is going to overcome step 10 of glycolysis. So I'm taking that OAA I transferred out, and I've got to make it into PEP. This is phosphoenolpyruvate. That's one of our intermediates from glycolysis. It was like right at the end. Remember, usually we take PEP and we use pyruvate kinase. And that gets us pyruvate. Instead, I'm taking that OAA and I went this long roundabout way through the mitochondria to get it out, and now I'm going back to PEP. And here's the reaction right here. We're taking that OAA, we're using energy in the form of GTP, and we're getting PEP. So that red phosphate group is coming from this energy molecule, and we see these blue carbons and oxygens leaving as carbon dioxide. All right, moving on. We're just going to keep doing lots of reverse. So that's how we're going to get pyruvate into glucose. That's how we're going to get pyruvate into glucose. But we said back here that pyruvate is not the only starting place. We also have amino acids from protein, and we have glycerol from fat. So let's take a quick pause. We're going to catch everyone else up that we just talked about. How do we get fats into this process? Well, what we're going to do is we're going to take our fats, we're going to break them down, and we're going to make it into DHAP. This is DHAP right here. And then that can go through gluconeogenesis too. So we started talking about beta oxidations of fats. We said that most of our fats have a glycerol, not most, fats in general have a glycerol molecule of some sort. And fatty acids. Sorry, just give me what am I doing? Excuse me while I mess this up. So this is our glycerol molecule here. I think I messed it up a little bit, but you're going to get the idea. And then I have my long fatty acid chains. So when we're talking about beta oxidation, we're talking about the Pac-Man chomping off of carbons from these fatty acid chains for energy, but we have this glycerol molecule left over. What are we doing with the glycerol molecule? We're going to put it into gluconeogenesis. So that's where this glycerol right here is coming from. We broke down our triacylglycerol. The fatty acids are off making energy through beta oxidation, and the glycerol molecule left over ends up here. So the first thing we need to do is use a glycerol kinase to make G3P. Your glycolysis alarm should be going off. G3P is one of our intermediates of glycolysis. And the enzyme name should be making sense too. Kinases move phosphate groups, so we just stuck a phosphate group onto this glycerol, giving us G3P. From here, we've got isomerases. This is kind of just normal glycolysis stuff. We can make G3P into DHAP. We can combine them together to make fructose 1,6-bisphosphate. So that's how we get fats into gluconeogenesis. All right, so now that we got fats, we're going to address proteins later. So we've got what happens to pyruvate, we've got what happens to fat. We're kind of now, either through special new steps or old reversible steps, we're kind of all landing here. We have two more special enzymes. The first one is fructose bisphosphatase. So fructose bisphosphatase is going to help us get back to fructose 6-phosphate. We can't use PFK1 anymore. It doesn't work reversibly, so we have a new special enzyme. That's all we need to know. Here's the new special enzyme. And the last special enzyme we have is this glucose 6-phosphatase. So remember, we used to do glucose to G6P using hexokinase. Not a reversible step. Means we need a new enzyme. We're also in the ER. Just remember that we're in the ER. What's the point of this step? The point of this step is to take that phosphate group off of the glucose molecule to make just straight up glucose. It can now leave the cell. Remember, when we made G6P, it was to keep it in the cell. Now we're removing the P so it can leave the cell. How does glucose leave the liver? The same way it came in, GLUT2 transporters. And remember, liver shares. Any gluconeogenesis we do in the liver, it could be for anywhere else in the body. The kidneys are doing the same process, but they're keeping anything they made to themselves. So, possible exam question, we undergo lots of gluconeogenesis in the kidneys. What happens to blood glucose levels? They're not going to change. That glucose that we made isn't leaving the kidney and going other places. It's staying within the kidney. It never touches the blood. And we'll address proteins now. So, we talked about this. Lactate to pyruvate, pyruvate through gluconeogenesis. We also talked about this. This is our glycerol from fats. We made it into G3P, which can be interconverted to DHAP. We can make that go through gluconeogenesis. I told you proteins also can undergo gluconeogenesis. They have their own special enzyme. So proteins, think back to when we talked about, I forget what lecture it was. But there was a long list of different amino acids and what they can be converted to. I think it was during the TCA lecture. There were a bunch of exam questions on it, like what can malate be converted to? That's why this was important. Alanine can be converted to pyruvate. Glutamate can be converted to alpha-ketoglutarate. Asparagine can be converted into OAA. These are all different molecules. Depends on what they are, depends on what enzymes they can use, but they're all going to make things that eventually can go to gluconeogenesis. So I kind of expanded some of these. Alanine goes to pyruvate. Pyruvate from here, they do like the same process that we just talked about. Alpha-ketoglutarate, easily through like one step of the TCA cycle, is going to be made into OAA. That's going to use the malate shuttle. We know how to get from OAA to glucose now. Same here, OAA, malate, OAA, PEP. Alright, moving on. So I said that we were breaking down fats earlier for this, right? We took off our glycerol, and our fatty acids were getting kind of munched off using beta oxidation. Hopefully this slide makes a little more sense now that we know some things about beta oxidation. We know that when we do beta oxidation, we're breaking off 2 carbon molecules to use for energy. What were those 2 carbon molecules? Acetyl-CoA. So, remember, multiple processes all happening at the same time. When we undergo beta oxidation, we're probably also undergoing gluconeogenesis. So our product from beta oxidation is that acetyl-CoA. That acetyl-CoA can help regulate gluconeogenesis. It's going to decrease pyruvate dehydrogenase, so that's going to decrease Krebs. Remember, pyruvate dehydrogenase was what made pyruvate into acetyl-CoA. If I stop that, I'm not going to be doing the TCA. I'm going to be doing this instead. The other thing it does is it increases pyruvate carboxylase. We saw pyruvate carboxylase all the way back in the beginning. Remember, that was how we got pyruvate into OAA, so we can transport out of the cell and make PEP. And same hormones as always. Remember, what does this hormone do? Okay, so how does it have to change the process? Glucagon wants to increase blood sugar. If I want to increase blood sugar, I need to make glucose increase gluconeogenesis. Insulin is always the opposite. So we're going to undergo glycogenesis. We're going to break down we're going to make our storage. We're going to, and we're going to make more different forms of storage, I guess. Triacylglyceride synthesis is making of fats. So we're going to increase this with glucagon, slow it down with insulin. They're always working in opposition to each other. I don't like that this slide is in here. I think it's kind of distracting. This has nothing to do really with gluconeogenesis. This is just telling us that glucose isn't the only sugar that we use. We can also use fructose. This is just an example of not using glucose, not undergoing gluconeogenesis. So if I eat an apple, there's going to be fructose in it. I don't need to undergo special processes. Fructose itself can enter glycolysis. Where is it going to do that? It's going to go straight to DHAP. It's going to skip that PFK1 step. I don't like that this is here, but it is. These are diseases. Remember, if I can't break down or store glucose, I'm going to undergo constant gluconeogenesis, and that's how we get these diseases. All of these diseases have this concept in common. So one of the enzymes for breaking down or storing glucose is broken, and so I'm undergoing constant gluconeogenesis, and I'm going to have some problems with that. Good things to memorize. And here's that drug we talked about, type 2 diabetes. Why do we get type 2 diabetes? It's not because we're not producing insulin, we're just resistant to it. Injecting a person with type 2 diabetes with insulin isn't really going to do much, if anything, at all. That's why type 2 diabetes patients usually have a much more diet-based control of their condition than type 1 diabetes patients. But they do have a drug option. It's called metformin. It's essentially going to stop gluconeogenesis, or the making of glucose. It's going to be just one way to keep those blood sugar levels lower, essentially. And that is all. We'll see what happens with my schedule. I might make a video on fats tomorrow. I might wait until next weekend. But hopefully this was helpful for you guys.